injury, quantitative MRI, the histology correlation, and then finally I'll show some results from different preclinical models. So spinal cord injury, it's a very serious condition. It happens mostly in relatively young individuals, otherwise healthy, and it results in a devastating change of life that actually affects these different individuals. So far, there's obviously no cure for that. With regards to the results of the primary injury in the spinal cord, it is the white matter damage that's usually the most serious condition, because it's responsible for the loss of function following this condition. Now, we know that MRI is very often used in diagnosis and general sort of imaging technique for looking at the effect of the spinal cord injury in the clinic. However, the standard clinical protocols that involves usually T2-weighted images and also diffusion are not very specific to them. So there's always a need for some more specific biomarkers. And that's what our lab has been trying to do for well over 15 years now. So with regards to the quantitative MRI, well, as we all know, quantitative MRI techniques basically just measure physical parameters that characterize the biophysical properties of the tissue. They have been used in the SCI studies, but mostly in preclinical models and preclinical studies. So today I'll be talking about three quantitative MRI techniques, which are particularly useful in looking at the SCI and the effect of the SCI. And these are two techniques that allows us to assess the damage to myelin, myelin water imaging and inhomogeneous magnetization transfer, and diffusion MRI that also provides some information about the myelin damage, but mostly looking at the axonal damage and sort of the general structural effects. So as we all know, myelin water imaging is essentially based on the T2 multi-echo T2 pulse sequence. It's a quantitative method. It was pioneered by Alex McKay here at UBC, hence our interest in using this technique in spinal cord injury models. And it's an indirect measure of myelin content. And there were numerous studies showing really good correlation with histology. So basically, if we look at, let's say, one pixel within our image in white matter, we can see that looking at the morphology of the tissue, you can see the multiple water compartment in this, and two particularly important water compartments. One is the water that is trapped in between myelin bilayers, and the second one is the intra-extracellular water. So if we acquire a multi-echo data, T2 decay data, and apply analysis technique, which is called non-negative least square fitting. Typically, when we do that, we actually use some kind of a regularization that we actually generate the T2 distribution. And we can assign these different T2 components to different water compartments. So the short T2 component is from myelin water, and the longer T2 component from intra-extracellular water. If you could just move this thing somehow. And potentially the long T2 component can be either from SCF or from some other sources of free water. So I'm having problems with the Okay, sorry. So the typical parameter that we actually extract from this analysis are myelin water fraction, which is the fractional volume of the myelin water, and it has been shown to correlate very well with the histology. So we started using this technique with regards to the spinal cord injury over 10 years ago. And here's some examples for in vivo and also the ex vivo scan showing the T2 distributions and the myelin water fraction maps. And we also have shown a very good correlation with the histology. Now, if we include both the white matter and gray matter, then obviously it's a bimolar distribution, so it will obviously correlate very nicely. But even when we look at the dorsal column white matter, it actually gives us a really good correlation with the histology. Now, another technique that can be used to assess the myelin content is inhomogeneous magnetization transfer. It is a sort of a modification of the older technique called magnetization transfer, which probably most of you are familiar with. So with magnetization transfer, we apply a saturation pulse on at a certain frequency offset from the water, and because of the magnetization exchange between the semi-solid pool and the liquid pool, the liquid pool is actually affected, and the signal is lower, and we can basically just calculate the magnetization ratio, which shows us how much the liquid pool was affected. And it's been used for assessing the myelin in many studies, although MTR is not a very specific measure of myelin. So in order to make it a little bit more specific, this technique was introduced, the inhomogeneous magnetization transfer, and it takes advantage of the fact that the structure of the myelin is such that it actually has something which is called a dipolar order, which is a spin polarization of dipolar coupled spins in the respective magnetic field. So this dipolar order can arise from the dipolar coupling, which is really the case with this bilayer type of structures and incomplete motional averaging in low spin diffusion. So essentially, when we have this semi-solid pool, the semi-solid pool now contains the Zeeman magnetization as well as the dipolar magnetization. And therefore, when we apply the saturation pulse, then the exchange of the magnetization will be from the semi-solid pool to liquid pool will be affected in a sense that there will be also exchange between the dipolar magnetization and Zeeman magnetization in the semi-solid pool. So in addition to that, we can see that the saturation is actually asymmetrical. Now, if we apply the saturation pulses on both sides of the water signal, then what happens is that the two components of the semi-solid pool, the Zeeman magnetization and dipolar magnetization from the dipolar order, are decoupled, and as a result, we actually get even stronger decrease in the signal in the liquid pool, and this difference between the one-sided saturation with dual saturation is this inhomogeneous magnetization transfer effect. So just like with MT, we can calculate the ratio using a similar equation. And here are some examples from our lab showing the HMTR ratio maps here on this side, and here there are some examples from the cord with injury in the middle, and you can see that indeed there's a very nice contrast between white matter and gray matter, and also in the lesion side we can see that the HMT ratio is actually lowered. Now, the one thing that we can do with this technique is basically just look at try to separate the marine water in the intra-extracellular water compartment and see whether there are differences in terms of the IHMT ratio between these two compartments that would tell us how specific to myelin IHMT ratio indeed is. So for that, we basically just using a combination of the saturation pre-module of saturation pulses with the CPMG readout. And it can be done either using a cosine modulator that applies essentially saturation on both sides of water at the same time, or we can use the pulses that are offset to the left and to the right of water. And then if there is some delay in between these two pulses, that actually generates a called T1D filtering. And here are some examples, again from the extracted, excised control spinal cord. And we can see that indeed the IHMT ratio calculated from the myelin water signal is much higher than in the intra-extracellular water, or for the matter, the IHMT ratio from the signal where both myelin water and intra-extracellular water are collected. And finally, the water diffusion. Again, I don't have to spend too much time. We all know that white matter in spinal cord is a very structured type of tissue, and therefore there will be different types of diffusion that can actually we can detect using diffusion tensor imaging techniques. So typically, the parameters that are used measured with the DTI is fractional anisotropy, which gives us some information about the structure, the longitudinal diffusivity, which typically correlates well with axonal damage, and finally the transverse diffusivity that has some correlation with myelin content. There are different ways of analyzing and interpreting diffusion data. Typically, they can be divided into two types of technique. The one group would be the signal representation, and the other one biophysical models. So in this presentation, I'll be talking exclusively about the diffusion tensor imaging, but we have used these other techniques in other studies as well. So the main reason why researchers would be interested in actually doing ex vivo experiments is that whenever the study is run, the MRI results are typically have to be correlated with histology. And that poses very often quite a bit of a problem. So the registration between the MRI and the histology data can be an issue, both with regards to the slice orientation and the slice location. There's obvious difference in spatial, very big difference in spatial resolution that may actually affect this correlation. And also the quantitation of the histology data are typically not a very quantitative data. In addition to that, the chemical fixation will always have some effect on the tissue microstructure, and it's important to understand what it is when we actually analyze and interpret the results. So with regards to the registration, so it's slice orientation and location, we started actually using in our lab sample-specific molds that are basically just modeled based on the typical structural MRI data and generated with a 3D printer. And I believe Kirsten will be talking about that a little bit more. Here are some examples of the molds that were created for extracted spinal cords. Now, with regards to registration, we actually developed a pipeline that actually allows us to get a very accurate registration between the two. In order to do that, we basically just use a multiple section of the stain and use a trace-weighted image from our diffusion data as a sort of a template in order to do the registration between different histology sections and also the MRI sections. And the same can be done here for.
Is it just me, or do we lose sound?
I agree. Yes.
Piotr, I think you muted yourself.
We lost him.
Yeah. Sorry, I don't know how it happened.
Got a little active with the space bar.
Yeah.
Good. Okay, good.
And the area chrome, which is the stain for myelin. Okay, now I have a problem. Okay, works. Sorry about that. So then we look at the sort of the difference in the resolution between the two. So the way to sort of overcome it, we basically just generate a grid which corresponds to the pixel size in the MRI data, and overlay this on the histology section. And then we use this Otsu method to basically just threshold the stain. And then calculate the area fraction, which is the segmented area of the total area, and in the end, that gives us the histology information registered to MRI, but also with the same resolution as the MRI data. Now, I mentioned the effect of fixation of the tissue microstructure. Definitely, well, we know that the tissue shrinks, but with regards to looking at myelin specific and using myelin water imaging technique, we were interested what else is actually happening. We actually noticed that the fixed tissue has consistently much higher myelin water fraction than the fresh tissue or in vivo. So we were trying to figure out what is the reason for that. So we basically just did a study where we have two groups of excised cords. The first came from the rats that were perfusion fixed with formaldehyde. And the other one was the cords where the animals were perfused with the PBS instead. And then these extracted cords were actually placed in a tubing, in a container, and we were basically just running 4% paraformaldehyde through this container. Over time, basically just providing the immersion fixation to both cords. And then finally, at some point, we actually left for both groups all the cords in the perfused and then repeated the final scan for 22 hours after excision. So we looked at the myelin water fraction, and as expected, we saw that at the beginning there was a big difference in myelin water fraction between the cords that were initially perfused with saline versus the one that were actually perfused with the paraformaldehyde, and we saw a steady increase in the myelin water fraction as this immersion fixation process was taking place, and at the end, the value of the myelin water fraction was pretty much the same for both groups. The T2 values didn't really change much for the myelin water. There was a slight increase. For the intra-extracellular water, it was kind of interesting to see a very steep drop of the T2 value initially, and then slowly increase following the trend of the saline perfused cords. this is the most important information that we actually got. So that basically just shows that the increase in myelin refraction is because of the increase in the myelin water rather than the decrease in intracellular water, which we initially expected. The intracellular water didn't really change all that much, whereas the myelin water definitely increased with the fixation. So, as I mentioned, we saw these changes in the T2, and we basically just concluded that the increase in the myelin refraction was to increase the amount of myelin water. And we speculated that this is potentially due to aldehyde creating artificially loosened portion of lipid bilayer. So there was a paper published in 2016 that actually compared two ways of fixating the white matter tissue. And the one was the typical chemical fixation, which is used pretty much regularly. And the other one was high pressure freezing that actually doesn't use any chemical fixation. And you can clearly see that the high pressure freezing preserves ideally the myelin sort of a morphology and the structure, whereas with the chemical fixation there are these areas where you can actually see the loosened myelin bilayers and the sort of gaps in between the individual bilayers. So we did a very quick and preliminary experiment where we used two cords, perfusion fixed, and one cord was perfusion fixed for 1 hour and the other one for 4 hours. Sorry, immersion post-fixed. And then we took the 300 micron section and then basically just they underwent the high pressure freezing. So the cooling rate was 40,000 kelvins per second. And then we prepared it for electron microscopy. So you can see here that the myelin period was different between 1 hour and 4 hour post-fixation. So definitely this fixation process affected the myelin period. The compact myelin, which was basically just this area that covers myelin, also increased at 4 hours post-fixation, so with post-fixation. So that indeed suggests that this process of chemical fixation basically just causes these changes in outer structures. Okay, so let's talk about the preclinical models. In our lab, we use essentially 4 types of preclinical models. The first one is so-called the dorsal column transection. It's a little bit artificial because it doesn't really show the actual primary injury type that actually happens when it happens in humans. But it's a very useful model, and I'll explain that in just a moment. The other models that we use are the ones that are more relevant to clinical cases, and these are the contusion, destruction, and dislocation. So with regards to the dorsal column transection, basically in this model, the entire dorsal column is extracted. Now, the dorsal column in rat spinal cord contains essentially three different types of tracts. The middle one is called the fasciculus gracilis, which is the ascending tract. The corticospinal tract here in this position, which is a descending tract, and then fasciculus cuneatus, which actually contains both ascending and descending tracts. So what happens if we actually extract the dorsal column transection, then we see a different type of injury in different tracts. And it's very predictable. So when we actually look at the position 5 millimeter, either cranial to injure or caudal to injure, you will actually see a different type of injury. So most of the damage done to gracilis happens on the cranial side or rostral, whereas in the corticospinal tract, most of the damage is done on the caudal side. And also the type of injury is actually quite different, because in addition to the actual damage to the axon on the site above the injury, there's a retrograde degeneration as well. So distal to the injury, we actually see a Wallerian degeneration, which is essentially complete axonal degeneration, a progressive myelin degeneration. It may actually take weeks or maybe even longer. And on the proximal side, we typically see the retrograde degeneration, which is progressive axonal degeneration that actually causes the retraction of the cut axons and also some bleeding. And here are some examples of the myelin water fraction map and how they actually correlate with the Laxalfaz blue stain, which is a stain for myelin. So here's the example of taken from our first study, where we actually looked more in details into the myelin damage and also the axonal damage from the dorsal column transection model. Now, the problem that we actually noticed in this study was that at 3 weeks post-injury, we often saw either very little decrease or sometimes even increase in myelin water fraction. And that was puzzling. And we basically just concluded that it must come from myelin debris that actually left following the injury. So the question was that, well, is myelin water imaging good enough to actually distinguish between the debris and the intact myelin? So we did a study looking at this specific case, where we looked at the control cohorts 3 weeks post-injury and 8 weeks post-injury, and we did both the myelin water imaging and the electron microscopy study. Going through quickly because I'm slowly running out of time. So basically, for the myelin water fraction, we took the region of interest that included only the pixels that were immediately adjacent to the sort of the central part of the fasciculus gracilis, like shown here. And similarly, for the electron microscopy, we basically just selected 3 pixels, 80 microns by 80 microns grid from the fasciculus gracilis. And we took the images at electron microscopy images at 16,000 and 35,000 magnification. So for the 16,000 magnification images, we basically we were trying to estimate the fractional amount of myelin seen here. So after using some denoising filtering and histogram equalization, we basically just manually segmented the myelin from here. Now, from the higher magnification images, we looked specifically at the myelin period, and that was basically just done at randomly selected myelin sheets within this area that was actually imaged. So when we look at the results, there was essentially no difference in measuring the actual value of the myelin period. And with regards to the myelin water fraction, we saw decrease from the normal to the 3 weeks and to 8 weeks. And the MRI results correlated very nicely with the results from the electron microscopy. Here's the Blan-Altman plot showing that there's essentially very little bias, and there was no trend with regards to an outlier, something like that. So basically, we concluded that the myelin water fraction correlates well to myelin content, regardless of the state of the myelin, whether it's a myelin, functional myelin or myelin debris. And here is an example of myelin debris, where we can basically just see this myelin bilayers that sort of collapsed. And so normally we can see that basically there are two types of water compartments in this myelin debris. So this is just the regular myelin water with regular myelin bilayers, but also these large water spaces where the water is actually more freedom within the myelin bilayers. So basically, just our conclusion was that, despite its inability to distinguish between intact myelin and myelin debris, it accurately reflects the myelin content. So we basically just tried to use a different technique, this IHMT technique with the CPMG, to see whether that actually may allow us to distinguish between the debris and the intact myelin. And the reason for that is that the IHMT is actually sensitive to the presence of the dipolar order, which will be more sensitive to the structure than the myelin water image. So these are the pulse sequences that we actually use for this study, and we use the T1D filtering at different level of the filter, which is changed by changing the distance between the positive and the negative frequency offsets. We did the 4-pool modeling with the dipolar reservoir. So we basically just look at the semi-solid pools, which was the myelin and non-myelin semi-solid with the associated dipolar reservoir, and two liquid pools, which was the myelin water and the extracellular water. And here are the results of the fitting and the parameters that we actually acquired from that. So we basically just did the analysis in the ventral white matter, which was not affected by the injury, and the fasciculus gracilis. And we could see that in the ventral white matter, there's essentially no difference between the control 3 weeks and 8 weeks for any parameters. Whereas for the gracilis, there's one parameter which was significantly lower, significantly different. And that was the T1D of the myelin semi-solid pool. So potentially, this parameter may actually allow us to distinguish between the functional myelin and myelin debris. It was a sort of a small study because it was done during the COVID, so there were some restrictions with regards to doing experiments. So right now we actually have another student who's working on a similar study with larger number of cohorts, and will be done at 9.4 Tesla, so with the cryoprobe, so we expect to have much higher SNR for this study. Okay, so just last section, couple of slides. So with regards to the clinically relevant spinal cord injury, I mentioned this two: contusion, dislocation, and distraction. Contusion is probably the most commonly used injury model. Dislocation is where there's a shift in between neighboring vertebrae, which actually results in damage to the cord. And distraction is where the two vertebrae are basically just pulling apart and stretching the cord. So that's the type of injury that is actually happening both to myelin and to the axons as a result of that. So we basically just try to figure out whether we can come up with some measure, MRI measure, that would actually allow us to distinguish between these three models, because that's actually important in a clinical setting, because depending on the type of measure, some kind of an intervention may actually be different depending on the actual mechanism. So we basically just look at these three groups of rats that underwent these three different injury models at three different temperatures. Now, in order to analyze this data, we came up, or I should say the MGU, who will be speaking later on, came up with this idea of basically just looking at the white matter damage maps and the damage occurrence ratio maps. So the white matter damage maps were basically just generated by looking at the histogram of the control pixels from the control cohorts, and basically just taking the value, the median plus or minus one standard deviation. And this is the example of the white matter damage map based on the longitudinal diffusivity. Now, the damage occurrence ratio map was basically looking at how often a specific pixel actually is damaged from the group of the chords for the same location and the same type of injury. And the same time post-injury. So damage occurrence ratio is essentially the number of times that this pixel is classified as damaged, standardized to the total number of chords. So here's the main result of this. We can look at the contusion at different time points, at different locations, dislocation and distraction. And just by looking at this, we can see that there are indeed quite significant differences in the severity, in the location of the damage, the timing of it. We can see very nicely how this injury actually spreads over time in the contusion case, and it spreads mostly to the cranial side and mostly in the dorsal column and so on. So so basically, we believe that we have shown that DTI can indeed be useful to perceive differences between primary injury mechanisms, and from the three DTI parameters that we looked, it looked like the longitudinal diffusivity was the most robust. And that's all I have for today. I would like to acknowledge all the people involved in these different studies and the funding of sources of funding, and we'll be happy to answer all the questions. Thank you.